the vitality of living systems. And it took me really a long time as a biochem professor and as a research scientist all this time to really start to comprehend. Complexity, the word itself doesn't even really come close to what it is.
It
It's
sounds like you're talking about the
mirror.
magnitude of the job. I mean, if we're going to put it into very simple terms, that a geneticist might look at coding, they might be interested in what particular region produces what particular protein. But what you're saying is, yeah, but that happens through chemistry. Chemistry inside a living.
It's all biochemistry. Yeah, those nucleotides had to be synthesized
Right.
and end up in a sequence, right?
And that chemistry is so complex and has to be on such a massive scale and timed so perfectly and have such great fidelity that when you look at it, it's almost awesome to behold, like looking at the Grand Canyon, because you have this incredible.
Like I say, it's like living systems are really a miracle.
Welcome to the Mind Body Literacy podcast and videocast. I'm your host, Nathan Schechter. A lot of the best sharing and learning happens in conversations, often ones that are informal and private. But what if you could be a fly on the wall and listen in? The Mind Body Literacy VP cast highlights different people with different perspectives. It's particularly aimed at sharing insights between professional silos of knowledge that work with the mind and the body. So, for example, massage therapists can hear how psychoanalysts think, and neurologists can hear how their ideas might be used by dancers or yogis. Because the mind and the body are too complex for any one person or group of people to understand it alone.
The information and content provided by Mind Body Literacy and in the Mind Body Literacy podcast and videocast is general information, and it's intended for educational purposes only. Individual situations vary, and this content is not intended as, nor should it be used as a substitute for professional, medical, or psychological advice, diagnosis, or treatment. No guarantee or warranties are given with respect to the accuracy, applicability, fitness, or completeness of the content.
With over 40 years of experience in biochemistry, Dr. Daniel Wera has worked at the intersection of research, teaching, and consulting, making science accessible to diverse audiences. Dr. Wera acts as a bridge between scientific research and real-world application, ensuring that the right people receive the appropriate level of information. His work covers a wide range of topics, providing in-depth analyses of published, evidence-based, peer-reviewed research literature and clinical studies in areas including biochemistry, genetics, biomedicine, pharmacology, microbiology, complementary and alternative medicines, nutrition and diets, and physical exercise for the promotion of a healthy population. His research and consulting have helped shape medical treatments, guide research initiatives, and inform healthcare decisions. In his consulting work, Dr. Wera analyzes scientific literature at various levels for different audiences. For academic and private industry clients, he focuses on research development and securing funding. For doctors and healthcare providers, he breaks down complex research on pharmaceuticals, supplements, and medical treatments, explaining how they interact with the body based on factors like genetics, age, and health conditions. And for patients, he provides consultations and clear summaries of the latest evidence on their medical concerns. For institutions, he conducts deep reviews of biomedical research, including cancer studies and clinical trials. And finally, for students, he offers guidance on cutting-edge research in biochemistry and medicine, helping them understand both animal and human studies. He also prepares professional students for exams like the MCAT and medical boards. Dr. Wera has also taught a wide range of biochemistry, molecular biology, and medical science courses at both the undergraduate and graduate levels. Some of the courses he has taught include graduate biochemistry, lipid biochemistry, graduate research methods, biochemistry, genomics, and molecular biology of exercise physiology and nutrition. You can learn more about Dr. Wera's work on his podcast, available on Apple Podcasts. It's called Authentic Biochemistry. Please join me in welcoming Dr. Daniel Wera. So welcome everybody to the Mind Body Literacy podcast. And my guest today is Dr. Daniel Wera. And he has over 40 years of experience in the field of biochemistry. And Dan, thank you so much for being here. I really appreciate it. because you do bring such a depth and breadth of knowledge in so many ways. You've published over, I think it is 40 papers. You have your PhD in biochemistry, and you also do many interesting things. You're the host of your own podcast, the Authentic Biochemistry Podcast. You break down for many different kinds of folks at many different levels, peer-reviewed research in the field of biochemistry and genetics. And you're also somebody, who's, very interested in finding the truth of how things really work and sharing that with folks. So it's really a pleasure to have you on the show. And I wanted to start us off just by talking a little bit about, models and ways of looking at things, because I think, it's a little tricky because, here you have, models of complex systems. And as, you've shared with me, so many events are happening in a living system, and trying to map that. I think you said to me once that you could go mad trying just to map what goes on in one cell, and yet models kind of aid our thinking, right? They help us solve problems, but it's also easy kind of to fall in love with a model or misapply a model or misunderstand a model. And when we can get this opportunity, to talk with folks and get a more nuanced sense of things, it does help us to get an increased likelihood of success. It helps us understand the world around us better. And it also, I think, gives us a better sense of sort of the excitement of learning. There's a lot that's unknown and there's a lot out there to discover. And one of the, key places that happened for me when we were first speaking was you just said something in the midst of our talking, and I want to share what it was and then ask you about it, but also break it down a little for those of our folks who aren't maybe, scientists or folks in the healthcare fields. But you just said to me, just in passing, you said, there's a synchronization between metabolism and gene expression. And you said it was, it's on the level of modifying DNA and nucleotides. And I started thinking about that because in my, journey and path, I had come along this way, where first I was looking at things from the perspective of exercise and nutrition, and then as I went deeper into it, it was, okay, now I understood about metabolic pathways, but I hadn't made in my mind, even though you begin to learn that exercise changes mitochondria and vascularization, and it does all these things, I hadn't really stopped to think about that bridge and that connection to genetics,
Right.
And then when you said that to me, I was like, oh yeah, it's not happening by magic, right? And you said it's transcription and translation. And so you really completed in my mind, almost like, I had gone and thought of having a trip. I had bought the plane ticket, but I had forgotten to board the plane, And
Right.
so like the process wasn't finished. And so when you said that, it really filled something in for me. And so that's why I want to start there. And just to, for folks maybe who are starting at a very beginning level, just to say, metabolism, which is this process of breaking down molecules in food, the major, groupings of food that we often talk about, which are lipids or fats. We often refer to them as, or we refer to them as proteins, carbohydrates, and then how we derive energy from these, and the way in which this gets studied, and then how that connects to what I even have learned just, in talking with you and then going back and looking at things that my understanding of genetics is very outdated, that these models of, genetics is a code that produces X is very simplified as a model. But this you started to share with me and you said, look, it is that there is this synchronization, that is something where the DNA is getting modified, the nucleotides are getting modified, that this is happening quickly, it's happening in real time, and that your metabolism is influencing what's happening in the genetics. Can you talk a little bit about that and maybe about these things like covalent, modification and molar concentration and frequency modulation? Share with the audience what that's about, how that works, and what that looks like.
Sure. Well, when you think about the living system, particularly a eukaryotic cell that is already complex because there are individual organelles subcellularly, and each one of those organelles are going to have membrane around them, which means immediately you understand that whatever's occurring in that organelle is unique to it. But you also understand that those organelles, none of them are working in isolation. In fact, for the cell to function, they have to be not occasionally, not randomly, certainly not randomly, and not even intermittently in communication. What's going on inside the lumen of the endoplasmic reticulum, or the peroxisome, or the Golgi, or the mitochondrion, or the nucleus, all have to be communicating for that cell to function. It's not simply that something has to change in the cell and then the mitochondria is going to, let's say, increase ATP synthesis because there is a need for more bioenergetics because the cell simply, the simplest mode you can think about, is ready to divide, right? So the cell could go through cell cycle, and that means two things have to happen front and center. You have to replicate the genome, and you have to replicate the membrane, all the membrane. That means de novo fatty acid synthesis, de novo cholesterologenesis. So the two major types of lipids, prenolipids and acolipids, those are two huge pathways. And for you to be able to coordinate that with the nucleus, you meaning the cell to be able to coordinate just at the biochemical level, means that all of those systems have to be poised at their correct molecular and equilibrium dynamics. And by that, I mean all of the intermediates in the pathway and all the potential enzymatic reactions, which can run into first there may be hundreds, then thousands, then tens of thousands of things have to happen. All not just like immediately, but in a particular sequence that is both spatial and temporal. So a lot of people think of the physicists coming up with space-time. No, it was the ancient Greeks that really understood space-time, and it's just we've been derivatizing that all the rest of our generations. So in a living system, you have functioning temporal events. And those temporal events are following a very, very, very specific set of time intervals. And okay, one in particular, when a protein is synthesized, first of all, you have to have nascent transcription. So that means proteins from the cytoplasm and maybe from one of those organelles I mentioned a few minutes ago, acting as proteins, have to translocate to the nucleus. Now that in itself is a temporal and spatial event. So it has to move, and those proteins aren't just like swimming in an aqueous solution. The nucleus isn't just, okay, everything is just water, everything's water soluble. So you have to think down to that level. In fact, transcription factors, which are polypeptides, they themselves also are mobilized with carbohydrate and with lipid. So it's not just a protein coming into the nucleus, binding to the DNA at a very specific location. We call it promoter and enhancer region. Those are called the cis-acting elements. Those are DNA, right? Your protein is working in what we call trans. So you have a protein binding to DNA. And yes, the protein recognizes that DNA, that huge macromolecular structure, as substrate, that simply put. But how does it find it? And how does it know where in this humongous genome does it find the promoter just to cause the expression of, specifically because we're talking about a cell cycle, acetylCoA carboxylase? That is the first enzyme that has to be expressed. First transcript made, then the transcript has to be spliced correctly, and there are splice variants. Then that messenger RNA has to be 5' capped and 3' polyadenylated so that mRNA now has the correct signature in a sequence. It's not simply a molecule. All these things we're talking about are events. This is real time happening where there are transformations of molecules.
I want to ask you a question about temporal, but before I do, I just want to translate some of what you said into layperson terms. It sounds like you're talking about a very complex coordination that, if we all go back to high school cell biology and we look at a cell, and inside the cell is an aqueous solution, the cytoplasm, and inside of that are the individual organelles, which each have their jobs, that the organelles have membranes, the cell itself has a membrane, and that these are not totally separate things. They are coordinating with each other in a very complex way. And then when we're talking about processes like inside, again, just to go back for folks to basic high school, inside the cell, you have the DNA, you have the strands of DNA, the two twisted helixes, and they're spread out like spaghetti inside the cell. And when the cell decides it wants to, you gave the very simplest example of divide. We have these processes of transcription where the two strands kind of separate a little bit because certain enzymes come along, sort of like if you think of running your hand along a bead of, a beaded, necklace. And as it does that, it builds up a messenger RNA, so a separate strand.
Now, when the genome replicates,
yeah,
that's a totally different process than transcription. Transcription is simply making a messenger RNA,
right,
particular molecular species, that when it leaves the nucleus, then will either be translated on polyribosomes in the cytoplasm,
right,
ribosomes in the endoplasmic reticulum.
Right.
And the molecule itself, the RNA, has a chemical signature, and that chemical signature directs it towards the polyribosomes in the cytoplasm or the ribosomes in the endoplasmic reticulum.
Right, it travels out into the cell.
So replication, though, is a totally different thing that DNA does. So DNA replicates, it recombines, it repairs, and it becomes transcribed. Four different molecular, all of them have to be coordinated for cell cycle. So new transcripts have to be made, but at the same time, the entire genome has to be replicated. That means that the DNA has to be replicated, every single nucleotide, and with huge, precise, pristine fidelity.
Right. So you're talking about massive things that are millions.
The daughter cell has to have the same genome. You can't the cell just can't make a mistake here or there. Any mistake, that genome is faulty and that cell will not survive.
Right. So you're talking millions of base pairs that have to be flawlessly recreated in a sequence.
Fidelity. We talk about DNA fidelity. Yeah. And when you get a mutation, people know about mutations. Sometimes mutations can have no effect because most of the DNA does not code for polypeptide. Okay. That's only like 5, maybe less than 10% of all the DNA will actually result in a messenger RNA. Most of the DNA is what we call intervening sequences, introns. It's only the exons that get expressed. And so when we first discovered that, and I had professors who taught me, were the people who discovered that sort of stuff, right? So I was really lucky to have them as my mentors, because it wasn't that long ago we didn't know anything like this. I mean, Watson and Crick in the late 50s, just figuring out the structure of one type of DNA. Now we know all of this, what I call florid detail. So people, when they study it, you can't simplify it. That's what I don't do in authentic biochemistry. That's why it's called authentic. If you want to learn what biochemistry is, which is the foundation of living systems, and what's more important to us than that, and it's not a bias. You ask a geneticist, what's the foundation of living systems? It's biochemical processes, because everything that geneticists do is a biochemical derivation, right? They look at the DNA and they say, okay, here's the particular coding region for a protein. That's like the dumbest thing to think about. That's the simplest thing to have, is a code for something. But how does that all get arranged simultaneously? And at the same time, abruptly stop. When I say abruptly, how about at the level of a microsecond?
Right.
all
So
the
you're
way
talking
up to
about,
millisecond and seconds. So if you have a stuff, for example, if you're replicating the genome, there are proteins that bind to the DNA that control, they're called cell cycle checkpoint proteins. And some of them, when they have a minor mutation, those are the worst oncogenes. One of them, for example, P53. When you hear a term like that, the P stands for protein, and the number after it has to do with its size in kilodaltons. Okay? P53 is a protein, is 53 kilodaltons. See? But that doesn't say anything about the protein. That's all geneticists want to know, is if this protein has a mutation, what is it doing? It's allowing the cell cycle to proceed, make a division. And what happens when you get rampant cell division with a mutation? That's called cancer.
right.
See? So
So-
we have checkpoints during cell cycle at a microsecond level. If something is wrong with the way the DNA is being replicated, it's stopped by these proteins. Those proteins actually destroy the protein transcription factor binding to that DNA and stop it from happening.
Right,
It's
they're
like,
like the
no,
traffic
this
cop.
cell will not replicate, because replicating it, if it continues to replicate, it can not only just kill the tissue, it can kill the organism. See? So the vitality of living systems, and it took me really a long time as a biochem professor and as a research scientist all this time to really start to comprehend. Complexity, the word itself doesn't even really come close to what it is.
It sounds like you're talking about the
Sure.
magnitude of the job. I mean, if we're going to put it into very simple terms, that a geneticist might look at coding, they might be interested in what particular region produces what particular protein. But what you're saying is, yeah, but that happens through chemistry. Chemistry inside a living.
It's all biochemistry. Yeah, those nucleotides had to be synthesized
Right.
and end up in a sequence, right?
And that chemistry is so complex and has to be on such a massive scale and timed so perfectly and have such great fidelity that when you look at it, it's almost awesome to behold, like looking at the Grand Canyon, because you have this
Like I
incredible.
said, it's like living systems are really a miracle in that we cannot create life. We can't except by procreating. What does that mean? Life begets life. You cannot take something that isn't living and turn it into life. There
Right.
is no way to do that. Okay? No way.
I want to ask you another question, because I know you talked to me a little bit about
The
membranes.
question even goes back to, where does life come from? You see, I remember Crick had this hypothesis called panspermia. Crick, who was part of the original Watson and Crick group, he moved to California, left Britain, and he built some the first biotech labs down in Southern California, La Jolla. And he worked on a lot of stuff after DNA, right? After the structure of DNA. And people would ask him these questions. And he would answer them and saying, well, we don't really know where life comes from. So maybe there was some living matter on a meteor and it landed, and that's where life comes from. So even way back then, people realized that we don't really have any kind of theory of where life comes from. It's not just like all of a sudden the earth cooled and we had these gases and we had all these compounds that turned into amino acids. It's like rubbish. Okay? You don't get life like that. You get a soup of a bunch of organic compounds. And not only that, somehow life, you have a membrane. That's why, as a lipid biochemist, I know people think I'm biased and they don't like it. Other biochemists don't like lipid biochemistry usually, because we tend to take center stage when we start discussing this, because you don't have life unless you have a membrane. Okay, how do you get these membranes? You say, well, it just circles around and now it's us and them, right? Inside, outside. No, membranes are so complex, and they're not just barriers. What they are is the communication network. All those organelles we were talking about with all those different membranes, that endomembrane, I wrote a review article on the endomembranous system a couple of years ago, if you want to get a copy of it. And I was the first one to come up with an idea that the endomembranous system, I mean, I'm sure I wasn't the first to come up with the idea, first one to want to publish a paper on it. Because the endomembranous system is actually the communication network of life. So the membranes aren't just sitting there allowing channels to be formed. The membranes are segregating and sorting everything from gene expression at the level of DNA going to RNA, from the RNA splicing to the RNA translocation to the cytoplasm, to the ribosomes, which is ribosomal RNA and transfer RNA and messenger RNA and 40 or 50 different proteins and lipids interacting within, making one protein. But that's happening at the level of 20 or 30,000 different types of proteins are being made at the right molar percent, the right stoichiometric ratios. The mathematics of that is beyond calculus, and I know calculus. There's no mathematics that, how does that work? Like, it's already doing that. So we go stumbling in as people. You know, yeah, you get a PhD in biochemistry in my late 20s. And I go, oh, well, we know all this stuff. This was the beginning of gene expression changes. So I was one of the first people in the lab we were involved in the synthesis of a synthetic gene. So, and this was in a USDA lab in Peoria, Illinois. The lipid biochemists, we were all lipid biochemists. We synthesized, because it was a small protein, so that means the gene is small in terms of how many nucleotides, for acyl carrier protein. And we expressed the synthetic gene in E. coli, and it functioned in E. coli. Now that, you know, didn't get much press time because it was in plants and nobody knew what acyl carrier protein was, but we were able to tear apart lipid metabolism that way. And it was like in the mid 80s, right? And we thought, wow, we know so much. I didn't even start to become a professor then. I didn't start to become a professor until 1991. And when you have to teach, right? This is something I always explain to scientists, young ones that are in my lab, or young ones that I'm training, or that I'm consulting with. People say, well, I don't want to teach. I don't want to do that, Dr. Ware. I want to be a researcher all the time. You know what you're missing out by not teaching? You're missing out on understanding really whatever it is you think you're doing. Because then you really, when you teach, and maybe you've taught before, I'm sure people listening to this have taught, you have to know that. You can't just simply go up there and chalkboard like we used to use a chalkboard. Now it's a PowerPoint presentation. You can't just go blah, 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 because you know what you don't know, because you're always true to yourself, no matter how much ego a person has. And so what you do when you become a professor, if you're a good one, and there are a lot that aren't, just like any field, right? They should stay in research or they should just write grants. What you have to do is actually fundamentally humble yourself down and say, okay, I have to learn what this means. I have to learn what anometric structures of carbohydrates are. Why do I want to know this? I remember learning it in graduate school. I have to teach this, which means I have to own it, right?
It sounds like you're talking about a depth of understanding that comes from needing to teach that's a deeper level than if one is just learning passively. And I remember when you were talking about membranes, you said to me that actually they have a sequence, that we don't even fully understand, that, we know at least DNA, you've mentioned Watson and Crick, so we know there is, and others who've worked on that, but that there is a sequence to DNA. But you were saying, no, membranes have a sequence, but we don't even understand yet what the sequence is to those. Can you
say
explain
that
what you
about
mean by that?
membranes, people go, their eyes just explode. Other biochemists do. Like, I think I told you, my oldest son is also a PhD biochemist. And he says, what do you mean by sequence? When he was younger, now he knows what his father meant. Just like we have a sequence in DNA and RNA, that means you have, if you want to think of it this way, a beginning and an end and a middle, right? That's how it is. 5' to 3' in DNA, amino terminus, carboxyl polypeptide. And everyone who's a protein chemist or a nucleic acid chemist knows each nucleotide matters in the DNA and RNA, and each amino acid matters in sequence. It's not the composition, it's the sequence that causes the DNA to be then the replicon for the genome, right? It's all about the sequence. So that, again, drives away all that nonsense of substance ontology. These are events. Even just the DNA molecule is an event that has to be read, right? It's not just sitting there and like all of a sudden it spills out messenger RNA. It's being read by RNA polymerase II and transcription factors and proteins that unwind the DNA, gyrases and tomoisomerases, very specifically right there, right now, for this particular sequence of events. And not just that, other parts of the genome are doing the same thing with pure fidelity. It's so pure that when it goes wrong, the cell kills itself. That's called apoptosis.
I
So
heard somebody talk about like the way that people learn genetics or
way
used to.
beyond. And what I tell people too is it's not like, well, you must be then a well-trained older biochemist to get this. No, what you need is a person that knows this, who can effectively give that to other people, that knowledge base. And that's what we've been doing as human beings since pre-Socratic times, right? Before we even were doing writing, we were sitting down and outside and having conversations with people. The Socratic dialogues, it's the same thing. If you can get a person's attention, that's first thing. If you're a really good communicator, that's something you learn. Some people aren't. They're gifted inside their mind, but they cannot communicate. That's something you have to learn. If you can't do it well, you should stay out of it. But the most important thing of it, the most important, is for the student to work, to study, to really want to know this. And that requires them, as we were talking about before we started this interview, they have to be motivated. And no matter how good of a professor you are, to motivate something has to come from the other person. So it's always an I relationship, even when there's 500 people in the classroom. And so for me to be able to translate what I know requires I have an audience that's interested in it. And I don't feel ever like I am lording it over them because I'm teaching them. I'm glad to teach them, because once you learn something, whatever it is, like learning how to play the piano. When I was a youngster, I learned how to play the piano. I haven't played it in years. Learning that, think about how that makes you feel. You can sit in front of a keyboard, you can walk anybody's house and you can play Mozart. I mean, my God, everybody is entertained by that. And it's not because of you, it's because you have a talent. So that you're not doing it to make yourself something, you're doing it to give to others.
Yeah, it
And
sounds like you're talking about how both relationship is a bridge by which information transfers, but also that one begins to see the world differently when one, has a conversation or a relationship where one is exposed to ideas maybe that are a bit of a different model than one was seeing or used to or knew before. And that if those two things come together, the relationship, the communication, the exchange of ideas and the motivation to be open to new ideas, that then new skills and abilities and possibilities emerge. Am I hearing that right?
yes, exactly. And also keep in mind, the more you learn, and think about yourself, each of whoever's listening to this podcast, the more you learn, because you're honest with yourself, right? You can't lie to yourself. I mean, unless you are delusional, unless you have such a psychotic break from reality. And I've learned this, I mentioned it to you, I think, before. I worked with neuroscientists and neuropsychiatrists who were some of the people that went into prisons to specifically interview people that are in there because they are mass murderers or something like that, right? And every one of them, and this has been published, many people have published this stuff. It's in the regular book form where you can read it. Every one of these people, these really nasty people that are in prisons, what they say to the interviewer is they say, oh, well, I would never lie to myself. I learned how to lie to others so I can control them and, whatever it is they're criminals about. But no, you don't lie to yourself. So even people who are twisted in that way, we all want to tell ourselves the truth. If you believe something, that's the truth you want to believe. You don't want to believe a lie. And so what this does is communication that you get to have with people who are learned in the subject, really learned in the subject, is this, it's a transfer, but it's also an association between who has the knowledge and who is gaining the knowledge, and eventually an equilibrium is generated. So that the person that's learning, they're not going to have the entire knowledge base that this one person or another may have to give them, because it'll take a long time to get that. But what they'll get are the means by which to have that transcendental event occur.
Yeah,
So,
maybe I can give an example of exactly what you're talking about, which is I think you're saying that if you have curiosity, if you have some inner integrity of perception that you want to know more, that you're willing to, be critical in your thinking and evaluate things, that if you have the curiosity, that you may not go from A to Z, in an hour or 20 minutes or in a 15-minute conversation, but the path then is open to you. And, if I was going to put that into terms of what we're talking about here, I think that is, part of what, happened in my own thinking was that as I, began to understand, like, okay, wait a minute, there's something called frequency modulation, which again, for folks who may not know, but like if you just think of an FM radio, that there's a wave coming, through the air to your radio antenna, and somebody's imprinted information on that wave. Most people can understand that. If you say your cell phone, it's a different kind of wave, it's a microwave or it's a whatever kind of wave. But again, there's information traveling on a wave. Most people go, okay, I get that. That's not too hard for folks, right? And then if you say to them, well, now think inside your body. And inside your body, there's a communication system that's just like that. There's waves that are traveling with information, but now we're talking electrochemical waves. And depending on the frequency of those waves, they're sending certain signals. Now, somebody might not go from that to understanding everything about, how the entire system works, but it's one piece that somebody might grab onto and go, that's a really interesting idea. And now they're starting down that larger path. Would that be an example of sort of what you're saying here?
Yeah. The only thing I would change about everything you said is absolutely eloquent. The only thing I would change is I'm kind of into terms, right?
Okay.
Curious, for example, the word curious.
Yeah.
Plato hated that word.
Okay.
And basically I do too. You know why?
Why?
It's not that curious is bad, but
right.
When someone's curious about something, it means that they have enough interest that they want to get enough information about it that they think at the level of just apprehending it, like grabbing it. But you don't want to do that. You want to obtain it.
Right.
That's what knowledge is. So many people, and I've had them in my classroom. I really want to know something about biochemistry because my dad died of Alzheimer's disease, things like that. You get it all the time. They come in your office and they say, that's why I'm in your class. And they, they're not even majors in a science. I said, sure, you're welcome in. And they might not have the prereqs, and sometimes they'd have to take the prereqs for let them in the class, that kind of thing. But whatever. Curiosity isn't what we're talking about here.
Right.
You have to humble yourself to say, I really want to have knowledge of this. Because just having curiosity, what you end up with is an opinion.
Right, not a passing dalliance, not a passing interest, but a desire for a mastery of a modality is what you're really talking about.
Think about all the people that are successful, and I don't mean monetarily necessarily, because I'm certainly, to me, that's not anything I'm successful at. Nothing wrong with it. But think about people who are successful. Well, every one of them will tell you, all the people I've ever met, really big names in my field, in science, in biochemistry, and even people in metaphysics that I've talked to, what they'll tell you is the same thing I just said. How did you get to be this person, this knowledgeable? I work on it all the time.
For sure.
But right now, today, when we're finished with this, I'm going to be working on a lecture I'm going to tape tonight. And I'm going to be learning things doing that, right? So it doesn't mean that everyone can gain the same knowledge as everyone else does, right? So you may be really good in chemistry, but not so good in biology. Right. So you can become an excellent chemist, but you won't be a biologist, and you also won't be a biochemist, you see? Because a biochemist, we're the weird breed in all of biological and chemical sciences. I think I told you this, but as a biochemist, the chemists have to take a course that I would offer. I only taught graduate biochemistry. I never taught undergraduate. I never understood that, because baby biochemistry is like, what is that? It's like nursery school. It's not worth it. So I always told the dean whenever I got hired somewhere, I'm only going to teach graduate biochemistry or medical or pharmaceutical. The school's beyond having an undergraduate degree. And the reason for that is I'm not capable of doing that. I know my limitations. Yet I say that, and I told you this before too, I think, before we started recording. I would go in, I got 4 children. They're all adults now. But every one of them, the oldest is 38, the PhD biochemist like me. I would talk to his teachers, and I would say, hey, do you want to have moms and dads come in and say, what does mom and dad do for a living? Because I'd like to come in and I could teach what I do. I'm a biochemistry professor. By then I was already at the university teaching. And they say, sure, that sounds cool. And I can tell you, Nathan, every time I did that, those young kids, I would say starting at about 3rd grade, not so much 2nd, but at the 3rd grade, they would ask me questions that sounded as educated as a 50-year-old attorney who would be listening to the same kind of lecture. And I wouldn't dumb it down. And I would teach them things that were not simple, like photosynthesis, which is not simple. Now, we think that mammalian biochemistry is complex. I started out as a plant biochemist, and I'm so glad because plants are autotrophs. Plants have all these other pathways, plus the pathways animals have, much more complex. So I would go in this 3rd grade class and I teach them photosynthesis, the light reaction. So they have to learn a lot about wave particle duality, right? And they have to learn about wavelength and amplitude and energy pulses. And then they have to learn all the biochemistry. We call the dark pathway, all the darks of the Kelvin cycle. And these kids, then I would give these kids a quiz, the take-home quiz. And, they would come back and they would work on it, right? And I'd come back one more time in that semester, usually in the fall. And it was amazing how well they could answer those questions with no background. And you know why? Because they wanted to.
And it sounds like you're talking about the love of
Younger
learning.
they were. It wasn't about what is Joe going to say to me? What is somebody else going to say to me? I want to know this because that's how a young mind is. And your mind can stay youthful, right? Look at you. You're learning things all the time. You run this whole system that you're doing. My mind is youthful. I'm still fascinated by things I don't know. I love learning. I took Clerk Maxwell's original treatise, right, on the electromagnetic spectrum. And it's in English, right? Published in 1873. And I tore it apart. I took about 2 weeks reading it, looked at the calculus and everything. I learned so much reading that one treatise, right? And I'm thinking, I love the challenge. Now I own that, right? Now I understand. And that's the basis, not just of physics, but a lot of what we know about biochemistry, right? Because it talked all about how electrical current can go through a medium, right? First time it was ever put together in that one treatise. So to me, you're always learning. It's exciting to do that all the way to the day you die. And so
it sounds like you're talking about the love of the learning, that you actually incorporate the learning almost like you digest food, that you take it in, you assimilate it, you're trying to understand it, and in that process, it sort of becomes a little bit of a part of you that then informs your increased mastery of your field. Am I getting that?
Life becomes beautiful. Life becomes more beautiful.
Right.
When I walk in the forest, if you're walking with me, if you want to hear it, I'm just going to start talking about it. I'll explain to you how photosynthesis works at different levels in the canopy, right? And that causes a red shifting of what different wavelengths of light. And then there's a photomorphogenic effect. That's why these leaves are curling now in the morning and not in the afternoon. I remember that from my plant biochemistry days when I was, 50 years ago, right? 45 years ago. I remembered it because the professors I had were amazing. And they were as amazed by the phenomena as they were teaching it.
It was one of the things that I really picked up in talking to you was this love of original thinking versus just simple pattern recognition. That that's a useful skill, but that original thinking really is a different skill. And I'm just curious, with everything you've studied over the years, was there a moment that like completely changed how you think about biochemistry or how you think about life? Can you pick one maybe as an example?
Yeah, I would say when I first started getting into the epigenetics literature. So epigenetics is a covalent modification of many macromolecules, but primarily what people are talking about, because the word genetics is there, epi just means above. It means the sequence of events that usually involves some kind of covalent modification of certain nucleotides in the DNA or RNA. Covalent modification means like adding something, right? A covalent bond, something simple like acetate or a methyl group. And what that does, it doesn't change the genome at all, although it can leave an imprint. We call it plastic, right? It leaves it because there's something called the methylome and the acetylome, right? They can be erased, by the way, next generation or maintained. Maintenance methylation. At any rate, what that does, that this is what changed my whole viewpoint tremendously. That's the communication from the environment that's always happening. And the environment, by that I mean everything inside the living system and everything outside of it. Everything is happening now. Like right now, when you're learning something, your central nervous system is changing the wiring of the associated synapses in such a way that you create a memory. And you create that memory and you superimpose it, you increase its density, actually more arborization around those particular axons. Well, how does that happen? So that you don't forget it, because you want to remember it. You think about it, you sit down with a piece of paper and you write out, what was this about? You go to any lecture, it's the same with me, right? I go to a lecture in subatomic particle physics. I'm doing the same thing. I'm taking notes, I'm learning. So the point is, we don't give ourselves enough credit as human beings. There's so much we don't have to turn into something that's magical. There's nothing magical about this, but there is something that's way beyond simple physicalist understanding. Living systems absolutely blow off the top from a physicalist perspective. There isn't any absolutely, here are the principles of life. We can give you 1,000 of them, but they're not principles. All they are observations.
Would you say that after all your years in biochemistry?
You a readout that's exact every time, and you can even put in a change in the code so it can allows it to be itself to be able to nuance around that code enough to be able to pick up something or lose something. But the living system doesn't just do that stochastically or occasionally. The cells in your body have the memory of everything they've been through at the biochemical level. And some of them you've been carrying, like the neurons in your brain, a really long time. That's why you have a memory of self. So the way you really get to understand living systems is you not just have to read science, you have to read fiction. And for me, it was reading philosophy, metaphysics in particular, but also a lot of epistemology. What were all these people talking about? All this metaphysics, right? People discard it now. We'll say, we don't really need metaphysics anymore. Now we have science. That word is ridiculous. Science simply means knowledge, scientia, right? Science you mean research science, right? And all that is a series of methods. But you have to expand your horizons too. You have to be able to, like recently I was reading The Idiot by Dostoevsky. I've never read it before. One of his books, I read a lot of them, I never read that. It's like amazing to read the talent of a real novelist, right? An amazing that was writing over 100 years ago, and he was Russian, right? I don't know what his culture was, right? How do you get that and then assimilate that into who you are? You read it carefully. You look at the way the words are put together, and it just blows me away. That's why books like that are amazing. That's why books like that used to be, you must read certain books. Like when I was in high school, we had to read some Plato, Socrates. We had to read the Republic, and then we had to dissect it, and we had to explain it. That was like junior year in high school. They don't do that anymore. And Shakespeare, we did a lot of Shakespeare, right? I mean, so much. And I remember, I don't want to do this. I want to do science because I always like science. So when you say what things have really turned me on, just about 3 months ago, The Idiot by Dostoevsky totally changed a lot of different, it tweaked me in a way that never had affected me before. And then I found out, I mean, this is an aside, but in the book, the character talks about a character of someone he's explaining about that said, what is it like to live in a place like a prison, knowing you're going to be executed, and then go all the way up to you're in the firing squad, and you're the next one to get killed, and then, no, by the way, we're not going to kill you guys today. And then 2 weeks later, you're freed. It's in this book, right? And you know what? That actually happened to Dostoevsky. The Tsarist regime was going to kill him, and the last minute he wasn't. And he was in line with the guys that he saw get killed. What kind of effect was that having you? And the way he said it in the book, putting into another character and just explaining it to other people on a train, it was like, that's what real knowledge is, experience. So your experiences all through your life are going to modulate epigenetically how you learn and how you interact with others.
And so it sounds like you're making a parallel, that you're saying, like, when you have DNA and enzymes are coming along, like somebody running their hand along a pearl necklace and saying, I'm going to touch this part, I'm going to touch this part, I'm going to touch this part, and these are the parts that I'm going to express. That's the scientific biochemical way in which we talk about it. But then you're saying life and ideas and experience can be looked at the same way. That you could look at that in almost a similar metaphor, even though it's got nothing to do with chemicals or enzymes or polymerases or, transcription factors.
But it does. But
you're saying the two are sort of parallels to each other, and that if one, and what I also hear you saying in a lot of things that you say is that if we think about genetics in a very simple way, like this causes blue eyes, this causes this, then it's a simple tic-tac-toe game that anybody can play, and it's very simplistic. But if we look at the greater epigenetic complexity, which has been described by people like Waddington and others, which is like a much more like anything could possibly happen, like when you drop a marble and it bounces all the way down and you don't know where it's going to go. Now you have a much vaster, complex, in all these ways that you've tried to describe today, possibilities of events that might happen that are so sophisticated and so vast that it almost becomes hard to say this causes this in a simplistic way or that causes that. And when you're confronted with that, what really takes the day is a better ability to think with original thought in a way that really begins to accrue mastery, because then you have a better shot when you throw the dart of coming close to the bullseye, at least, because you're not just repeating some simple pattern which doesn't match up to the complexity of living systems and how they actually unfold. Am I hearing that right?
That's exactly right. And again, you then, this is something that isn't very popular too, but is very popular from, like, say, existential metaphysics. The individual is the person that becomes knowledgeable, not the group. Okay. So you could say, yeah, well, as a group, we work together, and the Manhattan Project, take something horrible like that. Not really horrible, it's good science. But no, each of those individuals contributed something that was unique to them. So what really changed me, another thing, when I was in graduate school, one of my advisors was she helped us write our research papers because she was an English prof. She worked in our department, she was part-time, an older woman named Martha. And when I first started writing, Martha would look at my writing. She said, oh, you write really well, but you're not expressive enough. I said, well, this is scientific research. Isn't it supposed to be very cold? She said, no, what you need to do is you need to put in the accent of who you are. And once I got that, I felt how to do it. And she would read and she said, yeah, keep doing that. That really helped me see things from a point of view of an English literist, She had the right idea that whoever decided to put her in there with this was amazing. And she didn't talk to everybody because most of the other people didn't want to talk to her. I don't want you to look at my stuff. Who are you? Right? Because it wasn't mandatory. You have to be open to those things. You know, if you get on a bus and somebody's sitting next to you, if you feel comfortable enough to have a conversation with them, do it. You may learn something amazing from that person. Or you may just feel good that you had a human communication, right? And you have to be careful, of course. But when you're in a setting where you have like, and that's what you're doing every day, like going to a university or now interacting in this kind of format, that's what we're here to do. We're here to learn from each other. And what is required, the onus is on you. The onus, as much as you want to take in, is as much as you want to contribute. You see? And you have to humble yourself to do that. And if you really want to learn biochemistry from somebody, you have to go in there and learn it and take the time to do it. And nowadays, with all of this, you know, press always talking about, what about vaccines? What about drugs for cancer? What about drugs for diabetes? And I get people always ask me questions like family and whatnot, right? People want to know, well, my doctor said I should go on this particular pharmaceutical. And they ask me, and most of the time, the first conversation I have with them, they say, stop, I don't want to hear anymore. Because they think that what I'm going to say is, don't take the prescription. And I say, look, I'm not a medical doctor. I'm not one that writes prescriptions. I don't want to do that. But I will tell you that anything you're talking about, any pharmaceutical, how about any supplement? Okay, supplements are big. You really have to consider, why do you think you need to take this? What is it you think? What are they telling you it's going to do? Oh, it's going to boost your immune system. What does that mean? And then, you know, the person's telling you, well, I don't really know. What does it mean? So, well, the immune system is either, you know, hyper-inflammatory, non-inflammatory, or hypo-inflammatory. Which of those elements do you think get boosted by this particular, you know, organic acid in this, you know, extract? What are they saying? I can tell you, I can look it up and I can tell you. And people ask me all the time, whenever something comes on. And I say, look, this particular thing they have, it's not going to kill you, but the concentrations of what they say is the biologically active component in there isn't sufficient to act at the level of a binding constant, a KD, to ever affect the molecules in your body. So you would have to take in kilograms of this in one meal, okay? And then you still don't know it's going to have that effect after digestion. Because what happens when you take anything into your body, it's modified.
Right. It sounds like you're talking a bit about the ease of belief, that some people just want it to be easy. So you can latch onto a simple model. And then if you latch onto a simple model, you just say, well, that's how it is. Even if that's not really how it is, or it's misapplied, it's just easier than doing the work to understand something more. And that you run into that a lot.
And it's sad because people really do want the knowledge. When I talk to anybody, you know, people, I think I told you, people come in my office, years ago, a guy would come in and dump your garbage out, right? They would come by your office, you're in there late. I would have conversations with these people. It was amazing what their histories were.
Well, because do you think that this is one of the ways that it happens? That if people have more of a day-to-day relationship, they see somebody, they come in, they empty the garbage, they stop and chat for 15 minutes, they're there next week, that slowly the information starts to come out because it's happening sort of informally over a period of time. And that's another way for people to learn besides just sitting down in a lecture. I mean, both are useful, but that perhaps if people are often stumbling against feeling either overwhelmed by information or it's too much or they don't want to take it in, that when they're talking to somebody, that you can get these little, so for example, you just give the idea of a threshold. Hey, you're taking a supplement. Hey, does the chemical that you're putting into the body rise to a threshold? That's an idea that somebody could walk away with in 15 minutes. And also they could relate it back to what you were talking about earlier, which is, oh, you mean the things that I eat, they cause these substrates that cause certain thresholds, which then trigger things in genetics. Oh, I'm starting to understand this idea about how thresholds have to be achieved inside cells, inside the
Sure.
body, whether it's a medication, whether it's a normal metabolic process. And so they've learned a little something from you, even though they might not have the depth of somebody who's been at it at this intensity for 40 years. Do you think that's a possible way for people to begin
Yeah.
to travel that road?
What you're building there is the faculty of the understanding. Faculty of the understanding, the rational faculty of understanding, is populated by concepts. And concepts are abstractions, such as the word threshold, right? Or density, something like that. And people can get that. But equally important are ideas. Now, ideas are the instantiating real events. So concepts are abstractions. Like when you say tulips are beautiful, you can't say that and say that's actually that you've seen every tulip, have you? No. So you just say, well, tulips are beautiful. But what you can say, this flower I'm holding is beautiful. And then I'm going to extrapolate and say they all are, but really they're not all. Some of them aren't very pretty. Some of them are dying. Some of them haven't bloomed correctly yet, right? So you select, right? So you have to cultivate the other faculty, and the other faculty of the mind that really is neglected is the faculty of the imagination. And you imagine all the time. You imagine, you can put right now in your mind while I'm talking, you're listening to me, what the color of your dashboard is in your car, right? It's not there, is it? All imagination means that you can conjure something in your mind that's not there. Doesn't mean it never is there. Doesn't mean it's purely fiction, right? And so you have to imagine all the time because imagination is populated by ideas. And those ideas
imagination is a faculty of reason, you said to me. Imagination is a
Yes.
faculty. What do you mean by that? Imagination is a faculty of reason. What do you mean by that?
Your reasoning skills require a constant ability to take in information as it's coming. And when you're doing that, when the actual signal transduction cascade works because of the wave particles hitting receptors that are moving through your eyes and your other four senses, that phenomena is being translated in your mind immediately. But it has no meaning until, and this is something we have no biochemical correlate for, but it's what reasoning does. I can explain what reasoning does, but I can't tell you there's any biochemical correlate to it. What you do is you take something like, you observe something specific and you say, that is a camel. Now I know because I have concept camel. That's a grizzly bear. That's a human being. That person, that's a woman. That woman has blue eyes. See? So those concepts are in your mind. That's one faculty of reason, the understanding. You have that in your mind, you build it up over time. The other faculty, imagination, is taking in raw sense data and having it be specific. That idea instantiates as it's moving through your senses. And then you marry it, harmonize it to the other faculty, which is the faculty of understanding. And then what do you have? Experience. Okay? So it's a way to describe what you do naturally. Now, what is your mind doing? Synapses are firing. Neurotransmitters are entering the intersynaptic space. Sphingolipids are trafficking, various kinds of neurotransmitters across that cleft and firing an electrochemical potential. That doesn't tell you what that was, does it? You never will see something like a pyramid because of those potentials firing. You can't ever take that electrochemical, all the data, and you won't get something like a movie of a person seeing a sailboat going in front of them. Those are faculties of reason. And all we know is that that's what we do. That's what metaphysicians talk about, you see. Metaphysics.
Yeah, and that's something I've also taken is that as I've talked with you, that that you come at it from these two sides. I mean, you can go very deeply into biochemistry and, every specific, biochemical interaction and how it unfolds and the timing of it and all that. But you also have a very deep interest in how we perceive, how we learn, how we understand things. And it sounds to me that, one of the words that you mentioned that kind of maybe links the two together, I'm guessing, is the word experience. That as one gains an experience, I've come to think of it more now as I look at things, rather than thinking it as, an exact causality of A causes B, rather a network of possibilities, of vast possibilities, but with experience, one might navigate better amidst them. So if one was a mountain guide, one might not be able to say, oh, Mount Kilimanjaro is always the same every day, and I can predict exactly how the snows are going to fall and when it's going to be an avalanche and what bear is going to be where when and how the wind's going to be blowing. Like, nobody can predict that. But I've traveled over Mount Kilimanjaro 50 times and I know the layout and I've lived up there for 6 months and I've done this and I've done that. So I think if I'm going to navigate my way up Mount Kilimanjaro, I can divine a path based on my experience, but I couldn't tell you
That's a good way to look at it. Another word I would give you, and I've used it already, and you probably thought I was talking about something like, New Age or something. The word transcendental is the word that's incorrectly used. Transcendental means the means by which it occurs. So a transcendental that's what those two faculties of reason are. They're transcendentals. The means by which I have reasoning is I have a faculty that can make abstractions and a faculty can observe individual events. And I can marry them in real time and know as I'm moving through life what I call phenomenological reality. Now you have to think about it. So you don't have to think about your mind doing that. It's doing that. Think about that. Think about next time you're just doing anything, try to put yourself in the fact that you are ideation is constantly happening. You're constantly seeing people crossing the street while you're driving. You know where the other cars are. You have all these things that are happening in real time coming in your senses. And at the same time, that's being married to your faculty of understanding. And you precisely know how close you can get next to that car, how quickly you have to move past to make that pass on that 2-lane highway. You're not going, now I'm calculating this. It is the means by which your mind operates. So one of the things I always tell students too is that wouldn't you want to keep that ability really pristine, healthy? Yeah, because that's also what helps people to realize when someone's lying and when someone's trying to defraud them or hurt them, right? Lie to them. Because it's not so much you live long enough, you notice it, that helps. But you're able to pick up on what are the abstracted equivalents of what you're picking up? And you don't have to give them a name, your mind is doing it. But the more aware you are of what you're doing in your every life experience, because people have worked out, these are people who are philosophers. They have no idea how the brain works. That system I was explaining to you, that's in the Critique of Pure Reason by Immanuel Kant. It's Kantian Metaphysics 101. But now, when you read that, if you get that book and read it, I suggest everybody sometime read that book, you're going to hate it because he's not a very good author, doesn't write well. But when you study it, you go, oh my God, this person's explaining something that I only just said, yeah, I thought about this, right? He's explaining what must be occurring. And his words, I use different words. He didn't use the words I used directly. But I got the ideas from him, right?
It sounds like you're talking about both not only science, but how we apprehend science, and not only life, but how we apprehend life. That there's two pieces there, not just one. It's not just simple models or even complex and vast models, but it's also the way in which we use our human faculty to.
And then it allows you to prune that. You might say, oh, actually, I've been carrying around this concept for a long time, and it's BS. Because now I'm trying to justify that concept, and when it doesn't, I just tell myself, oh, eventually that's what will happen. See?
Right. It's sort of a mental flexibility.
Justified true belief. Justified true belief, JTB, that's knowledge, definition of it. So you have to justify those concepts. And if you continue to refute the null hypothesis, like in generating a research paper, generating a research endeavor, always consider your mind can do that about everything. And you're not sitting there saying, now I'm doing this, right? You know that's what you're doing because it's the means by which. It's like the concept of a lens. You look at the world through a lens, and that lens are your five senses. So you realize that a dog has similar senses, but the dog isn't experiencing the world like you are. Neither is maybe your wife. See? Same senses, different mind. Different mind because you are an individual mind. Everything biochemically in you, if you want to get down to that, is unique just to you. And we'll never know what it is because we can't measure all of that because it's constantly changing. Right?
Yeah, it's pretty amazing.
So that gives you also the nuance to accept more about, be respectful of people. Even if you think, I just like what this person's talking about, but respect, not just, well, they have these experiences, but the more you learn, that's how your mind works, the more you can learn complex things, like how to take care of your health without being a nutritionist or a biochemist or a physiologist. Yeah. And you don't just say, I coined the word exojective. I throw it out there and say, well, that's what the experts say. BS. What do you mean the experts say? What do you mean they're experts? Where is their knowledge base deriving this from? You can obtain that from anywhere.
I want to thank you, first of all, for all of your time and also for all that I've learned. Speaking with you has been so helpful and so broadening of my own understanding. But before we finish, I was wondering if I could just fit in two last little, well, they're not really little, but two questions, because I'd like to capture them. I mean, one was something you said about age, that, age is really a failure of the regulatory systems beginning to decay, that because the cells are no longer synchronized. And you did mention about hyperinflammation and hypoinflammation. I was wondering if you could just talk about that, briefly, about this concept that age isn't just wear and tear, but rather it's a failure of regulation. And then the second part of that question, if it is really a failure of like biochemical regulation, do you think aging is something will ever slow down or reverse? So I'm curious what your thoughts are.
First of all, no, aging is, sometimes I call it a chronic disease. You're aging from embryogenesis to death. You're not just aging because you're 60 plus, okay? So aging is an event that continues throughout life. All the living systems we talk about, for example, the control over lipid modification and turnover in the peroxisome, all the membrane lipids have to be taken out of the membrane, and then new ones have to be put in that membrane very specifically without the membrane ever noticing it. So it's not like a manufacturing, well, we clear everything out and then we fill it back in. It's happening all the time. The processing of that requires at the same time, the immune system is surveilling essentially every cellular mass, looking for any minor perturbations. And that can be regulated by such thing as chemokine trails. Chemokine trails are like a gradient coming from some cell that's being damaged, like imagine a cancer cell. Chemokines or glycoproteins are being generated from that cell, and there's a trail. There's a lot of it at the source, very little maybe at the lymph node, but enough at the lymph node that the T and B cells move to that gradient where it's the highest concentration. And if they're natural killer cells, they arrive, they check out that cell a couple of different chemical ways, and then they kill it. That's what NK cells do. They don't mess around, not like a T cell, typical classical T cell, like a Th1 or Th2, or CD4, CD8 positive T cells or B cells. Okay, that regulation is so complex and intergressed into everything that's happening in every cell and every tissue. That regulation starts to break down. The processing of all that information simultaneously slows down as you get older and older because there's so many different events that have to occur precisely. Life is like a miracle. I told you, it's amazing we live this long with the vagaries of an oxygenic environment. Because whenever you have molecular oxygen, you can make reactive oxygen. Reactive oxygen is what? It's a free radical. It has unpaired electrons. What is something with unpaired electrons? They go back to chemistry. It's looking to get another electron. So it pulls the electron out of DNA. Mutates that nucleotide base, you see? So that you constantly be bombarded with reactive oxygen, and your body has all these systems to take care of it, like ascorbate vitamin C, like tocopherol vitamin E. Those are only a few things. You have glutathione, you have enzymes that break down reactive oxygen all the time. And you need reactive oxygen at the same time through life, because reactive oxygen is how the lymphatic and the innate immune system kill cells that are damaged. Just produce a lot of reactive oxygen and the cell kills itself. Right? So you have to do that. So all that regulation breaks down over time. There's no way that that isn't supposed to happen, because what does happen the longer you live, and we have this, you can see it every day. Why do we have so much neuropsychiatric disease like Alzheimer's and Parkinson's, particularly Alzheimer's? That's neurodegeneration. So body can stay very healthy, but the body stays healthy so long, the most complex organ in your body, the CNS, it can't keep up with all the loss of the regulation, all the slow movement of all the lipids that have to get into specific regions, subatomic nuclei, be able to trigger certain subatomic nuclei in the hippocampus so they exactly occur synchronously every time. That starts slowing down. And what happens? What shows up at those sites is more and more the potential for damage, and then it damages it. So we live long enough now where we're increasing in the population diseases that we never had. And it's not because of nutrition. It's not because of the modern lifestyle being so stressful. We're living longer and longer. And no, not everybody gets Alzheimer's disease. There's a genetic and an epigenetic component to it. But can we predict that? No. It's not stochastic. It's pseudo random, but it's not random.
I want to ask you also about another just issue, which was you talked a little bit about cancer, and you talked about this is when you were talking about sort of the reasoning sort of that you alluded to in one other part where we were talking about, well, why would you approach it? And again, for folks who may not be at all familiar with these things, when people think about something very simple like how bacteria function. Some bacteria like salt, some don't. Some like oxygen, some don't. So when you think about how to defeat something, you often think about what process would we use, to defeat something. But in cancer, you talked about
take away an essential nutrient and kill it.
Yeah, right. Take away the nutrient. So you talked about with tumors, that they have sort of a micro environment, and that there is there's sort of an adaptation process where they're in real time biochemically, they're sort of trying to be flexible in terms of what they use to survive. And then the reasoning about how you target a specific thing to try to knock them out or whatever. Can you just say a little bit about that? Because I found that was very interesting and eye-opening.
What happens with cancers, like first you have an oncogenic event, and people typically think it's a mutation DNA. Mutation DNA means a protein may not be made, or worse, a protein is made, but it's not the protein supposed to be made. It's wrong sequence. So that protein then can generate what's known as a proteinopathy and start aggregating. This is how A beta proteins, and for example, one of the components of Alzheimer's, you get a proteinopathy that causes then the induction of an immune response. Okay, so that processing of the tumor starts off being a cell like, say, a liver cell, hepatocyte. But then something happens to that cluster of hepatocytes, and all of a sudden they're not hepatocytes anymore. They came from there, and they have a lot of the same metabolism, but the metabolism has changed slightly. So they're not that anymore. They're a cancer cell. Not only that, the cancer cell then, because it's starting to generate a change in metabolism, like switching now to just glycolysis rather than fatty acid oxidation. And that means it can use glucose and make only a couple of ATP, unless it runs through the TCA cycle, electron transport chain, OXPHOS, much less than you can get from beta oxidation and fatty acids for energetics. So you might think, why does a cancer use glucose then? Well, because glucose is water soluble. So it can get it without going through the effort of binding proteins and the slow motion of lipid movement as an energy source. So what has that tumor cell environment already done? It's changed the metabolism, not just of the tumor cells, but it's communicating with everything around that's perfectly healthy. And it's not making them cancerous. It's making them deliver information that's different because that cell, which was right next to them in the tissue, which was a hepatocyte, communicating perfectly well to make that tissue, is now signaling the wrong signals. So those cells are not tumor cells yet, but their metabolome is completely wrong. So as this continues on, more and more cells start developing a phenotype, a biochemical phenotype. And what is measuring that all the time? The immune system. That's its job, to surveil 24/7 embryogenesis to death. That's all it wants to do. And not only do that, surveil, arrest, detect, decide, kill, right? All those things, right? You have all these different immune cells. So what happens in a cancer and tumor microenvironment, you don't just have tumor cells there now. They're not hepatocytes anymore. You also have immune cells in there. And you also have fibrocytes in there. And you may have endothelial cells in there. What happens now is all of those cells start signaling according to the tumor microenvironment. They start communicating, changing their metabolism, changing gene expression, all of which facilitates the growth of the tumor. Not because the tumor it's not a master controller. It's because if the tumor is growing, that's what happens. See? See how that fits into what? The transcendental. It is the means by which it's doing that. So we go in, we design a drug, we're going to target a specific sphingolipid, sphingosine 1-phosphate. Because every time we have this kind of tumor, multiple myeloma, this level of that particular lipid goes up, and we know what that lipid does. It causes what? Cell cycle. We got to figure it out. We're going to kill that. Well, what happens when you do that? What about the pathway of sphingosine 1-phosphate? It's a completely elaborated pathway, and you're destroying the production of that. Now you're going to change the pathway in another direction, spatiotemporally, in healthy cells, and even in the tumor cells. And all that happens is, as long as that cell keeps on dividing and dividing, it becomes a tumor. So the result is cancer, right? So it's the means by which it grows. So even when you add really sophisticated pharmaceuticals, or even monoclonal antibodies to specific proteins, or immunotherapy, eventually it breaks down. It breaks down. Eventually, those same things you put in there, even the immune cells, are enhancing tumorigenesis. It's in the literature. I'm not making this up. That's what the literature shows you. So, but the literature, people that are writing this really talk about one specific thing. We are interested in natural killer cells. We are interested in Th2 cells. And even if they're looking at all the cells and they're doing, where they use algorithms and they look at entire pathways, they don't know what the pathways are doing as they're changing in real time. You see, because they're constantly changing. And an individual metabolite that was a normal metabolite in, say, the Krebs cycle, the TCA cycle, now becomes, new term, an oncometabolite. So why is citric acid all of a sudden promoting cancer? Because it's being synthesized in the wrong subcellular compartment, or there's too much of it or not enough of it, or it's altering the level of isocitrate. And now you find isocitrate dehydrogenase isoform 2 that's in the cytoplasm that normally only carries out salvage pathways for amino acids to make fatty acids. That's one of the things that naturally occurs a priori. It's always there already. Now all of a sudden that gets more robust because more carbon's coming from the cytoplasm, all water soluble. And when that becomes more robust, that whole system becomes oncogenic. That's
So
what I'm talking.
maybe I'll close by asking you this. If the world is so vast, in so many different ways, from the ways that we perceive to biochemistry itself, to the multiple systems and cells and ways in which one thing influences another thing, how do we then move forward, in life, in a world where not everybody can be a biochemist with 40 years of experience, where not everybody can, but yet we all have bodies, we all have lives, we all have health, we all go through states of wellness and disease. What is a way, do you think, that we can begin to maybe not, grasp too tightly simple models, and hold on to them with a death grip because we just are fearful and need something to believe in, but yet not be overwhelmed that it could take us 40 years and we'll be at the end of our lives before we could ever make a decision or know anything? What do you think is the the middle ground for folks as they go through life. What have you found to be helpful in your own journey, and what are your thoughts about that, having seen other people maybe even outside the fields of science?
The answer is very simple for me. Think about who you are as an individual and try to do good and do better all the time. I'm serious. Do better. Not just say, I've done my best. Best, you can't do any better, you see. I get that actually from Kierkegaard. You see, you pick up things like this over time. Soren Kierkegaard, the metaphysician, writing 150 years ago. It was the first time I never thought about that. That's why terms are so important. When you think about always your health, okay, which I don't, right? You know, they used to say the medical students, they think they got the disease they're studying. That could happen, right? You know, you see a movie and you think, oh gosh, that could happen. I get a home invasion. It's the mind works that way. Safety. People are too hung up on safety, too hung up on security. Doesn't mean you don't want to protect your family. Of course you do. But think more about the quality of your life as you live. Don't be so worried about how long you're going to live. Think about when you're living, are you doing things that you feel you know are good? Good for yourself and, most importantly, your beloved, your family, the people you care about, because those are the people that are the closest to you. And when you do that, you're not so worried about things. And because of that, you're healthier. Because to do that means you're spending a lot of time with your mind and trying to say, hey, I don't want to act that way. I don't want to make this person upset. I don't want to choose this particular route, go into getting all this medical treatment, if all I know is I'm going to be sick for 3 more years. People start thinking about that, using their reasoning skills. They'll live a healthier life. And everyone knows this. You don't need any of this education. So we want to know a lot, and we do want to make, I mean, I would be glad to help people's lives better. But, you know, the kind of things I would tell them, don't drink alcohol. No alcohol, because alcohol is a toxin. You know, any amount of it, okay? No matter what people think, it's not good for you. Okay, that's one thing. Don't gain a lot of weight. Gaining a lot of weight makes you, it's aging at a much more rapid rate. Okay, things like that. Those are simple things, and everybody knows this. You don't need to, but they want, what people want is they want to live their lives and do whatever they want to do, and then they want to say, you know, we used to say this in the 60s and 70s, you know, that the adults would say this, just take a pill. Now, how about forget about that? Eat natural food, eat normal food, exercise. When you think you've exercised some, double it, triple it. When you think you spent a lot of time working on a subject and your mind feels tired, listen to some Mozart or some Beethoven or some Schubert, and a half an hour later go back and study for 3 hours. So I used to tell my students, and it works because you're controlling it. So again, think about when you're at a cliff, and that cliff is the abyss. You say, I can't jump across that area. You think about, you know, movies you've seen where that is. How do you jump across that cliff? Because you tell yourself you're going to, right? So you make it smaller and you run faster when you get across it. That's a metaphor for how you get through life. You know, you tell yourself, this is a journey, and you know in the end you're going to die. So don't be afraid of it, and don't hasten it, you know, and don't hasten it in others, you know, because every human being is precious. Everyone, even people you don't care about, right? And do what you can to do better, right? And that means improve your knowledge base. Start playing the piano, pick up a violin, you know, whatever it is. See, so don't ask me for advice, because that's my advice.
No,
The
I
thing,
appreciate it. I appreciate it. And I, again, want to just say I appreciate
all the biochemistry and all the knowledge that you want to have people listen to, that's all important to me, right? And so I love teaching it. I love it when people learn it, because then it's like what we used to say in the 60s. I'm turning you on to something, right? Life is more interesting to you when you walk out in the park or riding up on the bus, because you know more about what's going on. And why worry about the fact that, yeah, it's going to go wrong and one day your family will bury you?
Yeah, I think that's
that-
well said. That the other thing that you get is the interest and the wonder as you go through it. That anything can be,
always exciting, Nathan. It's always exciting.
yeah,
Even though you think you still have worries, who doesn't, right? And you don't like pain, and you're hoping that you're not carrying around a cancer that's, indolent, smoldering. But if you are, well, you're going to die. And if you die from cancer or you die from an autoimmune disease or cardiovascular disease, those are the three major ways you're going to go.
Yeah.
So, I just don't think about it, and also the other thing I suggest is what makes people get more courage. Take chances, even when you're older. Take chances about, experiences that you have. Why not? And again, bottom line, the categorical imperative, don't hurt other people. Treat them just as important as you are. Treat them as individuals, right?
Yeah.
Never as something that can do something for you. Why would you want to do that, right?
Well, thank you so very much. I really appreciate all you shared, and it was a pleasure to have you. And I hope perhaps some points we'll speak again. Thank you so much.
I really appreciate it, Nathan. It was good talking to you. You're a good guy, and good luck.
Thank you.